So our first parent speaker is Yiwei. Yiwei is a dedicated mother and advocate. Upon her son Leo's diagnosis with arterial neurodevelopmental disorder caused by a TMPO2 gene mutation, she took extraordinary measures to find a treatment. Despite being told that nothing could be done, she founded the TMPO2 Foundations, and through collaboration with biotech companies, they developed a personalized medicine, a personalized antisense oligotherapy for Leo within just a year. Encouraged by Leo's progress, Yiwei expanded her mission to assist other families facing similar challenges. With her background as a machine learning scientist and mathematician, through initiatives like Project Baby Lion, Yiwei aims to revolutionize the diagnosis and treatment for arterial disease in children, striving to make personalized medicine accessible and affordable for all. In a recent initiatives, Yiwei and I are exploring the opportunity to collaborate with hospitals and patient foundations from China through the large patient population in the country and China's investigator initiative trials, or IIT program. We are hoping to accelerate clinical practices with more flexibility. Without further ado, Yiwei.
Thank you, Nianwei. Thank you for that introduction. And it's a pleasure and an honor to be here speaking to you all. So I will be telling you about my son Leo's journey, and focusing on the part that everybody I hope is most interested in is how do we get a drug made for a baby. And as Nianwei said, it's a very collaborative effort. So these are just a small number, these are the leads of our teams, but there's a lot of individuals that helped in the effort. Our principal investigator, Dr. Nicole Colfell at Rady Children's. Oh, not this pointer. Okay, thank you. Dr. Colfell, Dr. Dimmick, who was the chief medical officer at Crayon, or former, and then Chris Hart, also the former CEO of Crayon Bio. And there were a lot of collaborators at UCSD as well. So here's Leo when he was born. And I will spare you the diagnostic odyssey because I think everyone here is very familiar with it. But there were signs at birth, and we got Leo sequenced when he was 3 months old, getting the results back when he was 4 months old. And the paper, so he had to be sequenced in a clinical trial. So sequencing and diagnostics is still not widely available or accessible. And this is a problem at a systemic level. But when the clinical trial for the diagnostics came out, there he is, the lone TMPO2 patient, in the study to kind of to find genetic diagnoses for infantile onset epilepsies. And the incidence rate, the prevalence is extremely high. So almost every baby, I think that the incidence rate is up now to 80 to 90%. So many babies with epilepsy have a genetic etiology. And so, that should be a first line tool when you see a seizure in a baby. So that set me off. So people told me there's nothing you can do. And that didn't seem to be true to me. And even if it were, I was not going to let it stop me. So there's also this thing that brain development happens, for young children, for babies and young children. And it's a very, very fast and dynamic process. And so much is going on from even in utero, but also after birth, all of these things are happening concurrently. So we were on a race against time, and our child, luckily for us, he was not terminal. He did not have a neurodegenerative disease. Nevertheless, his quality of life was on the line, and it was the quality of life for the rest of his, possibly 8-9 decades of life. So we wanted to make a drug for him as fast as possible. So there was a crash course in drug development, and this is what the process looks like. So there's the basic science, there is preclinical work, then there's clinical work, and this is the design of the FDA and the system that we have in nowadays. And then after phase 4, there's all of this commercialization stuff, which actually is extremely important for accessibility and equity. right? So for me, I thought, okay, what is this all for? I don't care. I just want a drug in his brain ASAP. But then, looking backwards, in order for, every child who needs one of these drugs to benefit, we do need to find a way forward for this step, and also, for every single step in this process. So how do you make a drug? Well, we found the cutting edge partners. So Crayon Bio is an AI first, drug discovery and development company. And with my background in machine learning and AI, I was able to read their patent and do the due diligence and have enough confidence in this company that had never made a drug before. But their patent looked, solid. And so we signed a contract with them and we started working together. And we also, started the whole system of model organoids. So this is the other half of the drug development process, is trying to find, a model of the disease in something other than patient, so that you are not taking the risk with the patient. And so we didn't make Leo yeast, we made Leo worms, we made Leo flies. Actually, Leo flies were already made, but we had to make, more faithful Leo flies. We made Leo mice, and we turned down a proposal for Leo monkeys. They were not only extraordinarily expensive, but then you have to, once you have the monkeys, you have to take medical care of the monkeys, which is also actually very, difficult. And the most promising, and then what ultimately succeeded, were Leo brain organoids. And so we were very, very thankful to the Motri lab and its collaborators. But these organoids, they come from iPSC, so they come from stem cells, and so the first step is to make the stem cells, which iXCells is a great a resource for. And on the other side, concurrently, we were making the leads and we were qualifying the leads, and we were going through, this is a standard workflow for ASOs. There's a different workflow for different modalities, but the shape generally looks the same. You start with some leads, this number might be millions for small molecules, or smaller for gene therapies, but then you select down, you try to design rationally when you can and engineer when you can, and apply AI when you can. And increasingly, AI is more and more applicable and effective at each stage individually. And also for ASOs, the company that we worked with, Crayon Bio, they could, out of 100 leads, they could select over 80 of them were safe in kidney and liver tests. So that is a very strong bias towards safety, even at the first step of the screening process. Our drug necessitated allele selectivity. That is a technical point I won't go too deep into, but we found a drug that could hit the target with a very beautiful, dose response curve. So this is what it looked like. This is the Gantt chart of everything running in parallel. And this was, we wanted Leo to have the best drug, as fast as possible. And there's a lot of failure. So there's a lot of hedged risk. So I guess, it's not, I don't want to say failure. I want to say, we mitigated a lot of risk. And so if things happened differently, we would have still gotten to, our end result within about the same time frame. And that is what a significant amount of resources were expended on. So ultimately, the disease model, actually, everything other than the top two is disease models. And all of these disease models, they all failed for different reasons, except for the the iPSC derived organoids. So we barely succeeded in some way. So this could have also failed, and then we would have had no disease model. And then the question would have, or the decision would have been, do we go into the human? Do we go into the clinic with no wet lab backup data and only on the back of some bioinformatic data? And that is, that is a harder question to answer than what we had to ultimately answer. And so here is, thanks to the Motri lab, this is the on the right-hand side, you can see that these are the Leo brain organoids. The vertical axis is the diameter of the brain organoids, and we see that his microcephaly phenotype is recapitulated in these brain organoids. And this is actually, these brain organoids are a very faithful representation of late fetal brain development. And so in all cases of microcephaly, which occurs with an incidence about 1 in 10,000, this is an excellent disease model, a place to try to find a disease model. And then our rescue data also came back to be very promising. So the red line is the control. So this is what happens to the brain organoids if you don't apply anything. And then we have 3 different ASOs at different concentrations. And we have 2 time points. And we see that ASO B in particular has very good performance because we are not only clearing the 50th percentile, there's a big jump in the 50th percentile, but there's also a big jump in the 25th percentile. And I think actually we got our dosing a little bit wrong. These 5 to 10 micromolar doses are a bit too high. Sorry, I'm jumping into the technical. I'm a scientist by training, but this rescue data was extremely promising and encouraging for us. And it came in January of 2023. So this was after we had the leads. Yeah. And ASOC, it was a control ASO. So this is just a scramble and a non, it's not supposed to do anything. And we see indeed that, it doesn't do anything. And in fact, it's a bit harmful at the highest dose. Okay. So with that, onto the clinic. Well, so between January, this is in January of 2023, and July of 2023, there is the 6 months of applying to the FDA. So, that is one of the problems in this space, is that we have safety data in non-GLP trials. We have efficacy data in patient-derived cell models, but it took 6 months for us to get into the clinic. And something about that risk-benefit, outcome doesn't sit right with me, because Leo was having just, sometimes up to a dozen seizures a day. And often he would stop breathing during these seizures. So there's a conversation to be had with FDA about how they regulate these therapies. And so here is our clinical protocol. We doubled every month during the escalation phase, again for safety. And then after that, we did every 3 months at the maintenance dose. This is again a science slide, but it's trying to find the pharmacology. So trying to figure out, are we hitting the target in the CSF, in the brain? And this is a assay that measures RNA in the CSF. So there's actually a tiny amount of RNA in the CSF, and it turns out there's maybe five molecules of the TMPO2 transcript. This was 200 milliliters of CSF. So there's a very minute amount, and we tried to measure it, and it's incredibly noisy. So, at one point there was 100% of it was the mutant RNA, and at a different point, only 25. So only like 1 or 20%, 1 in 5 of the transcripts. So this is, we tried our best to measure it, but it's very hard. And, this is one, indication of the challenge of trying to do these trials scientifically. Next slide. There. Okay. So seizure data also came out to be very promising. We saw the decrease in seizures, especially these dark, severe grand mal seizures right away. The second thing we noticed was that the seizures would recur about a month in to the 3-month maintenance cycle. And I think the issue there again is kind of it's both the FDA and our lack of pharmacology study before we went into the patient. One thing that helps FDA applications is if you hew to precedent, then they will approve you. One thing that does not help you is if you're proposing something new, and then you might get bogged down into a conversation. So we hewed to precedent, and we proposed a protocol that was, that had been submitted and approved many times. And so, this between these 2 doses, this is when we tried to have a conversation with FDA about increasing the frequency, which they did approve. So thank you, and kudos to them. So we are now dosing every 6 weeks, and we have been seeing increased, seizure control as well as developmental progress. I should skip this. And yeah, so the last piece is, the thing that we, as Leo's parents, thank you, as his parents, wanted most out of the clinical trial was not seizure control. It's 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 neurodevelopment. And so this is one of maybe the other half, or maybe the main challenge is how do you measure, childhood development scientifically at an N of 1, scale. And so some of the things we did, well, so these are the successes, right? So we got a new milestone, which is he went from not having head control to being able to sustain a tripod sit, which is, a significant improvement. So he can't get himself into the sitting position, but if we put him there, he can balance himself. There's less spasticity. This is both an objective and a subjective measure. So with AI and newer technologies, we can, have objective metrics for spasticity. He has new social emotional developments where he will laugh and cry with his sister. So the crying was the crying was very easy to notice. Before, I had one child crying at a time, and then all of a sudden, whenever I had one child crying, within 5 seconds, I had a second child crying, and it was very fun actually to have this experience because I knew what it meant. Strabismus, a lot of these children with neurodevelopmental diseases, there is some manifestation in the eyes. And this is another opportunity for an objective metric. And again, the developments in AI, especially these multimodal models, can make, a clinical measurement of this kind of a symptom, objective, cheap, and easy. You just have to tell the model what you want, and it will measure it for you. This is really hard, but we noticed, a big jump in responsiveness and presence. He went from a baby who sometimes, we would ask, like, is he in there? Is someone in there? And I know a lot of parents have this experience to a baby. We think, yeah, there's definitely someone there, and there's something he wants, and, and all these things. And in terms of there's something he wants, he will attempt to reach for toys. He will interact with toys. He will interact with sounds. He will notice cause and effect when he makes a sound. And, we are really excited about the new sounds that he's making himself. And since the increase in dosing frequency this past summer, we've noticed there's less regressions between doses, and some new sounds that are consistent. So I think, in terms of development data, in terms of clinical data for childhood development, I don't have a complete answer, but the tools are becoming available, right? So there are tools for getting biomarkers for EEGs, for blood sampling and other, biomarkers that you can take at higher frequencies. So there are, there are real, blood droplet tests, which you can send to like, say, the Snyder lab, and you can measure hundreds of different biomarkers in the blood. And there's now, dry scalp EEGs where you can measure, at a high frequency, at a high resolution, so that you can get objective data from a single patient, and it can power a whole trial from just a single patient. The data from a single patient, let me say it this way, the data from a single patient can power an entire trial if you take the measurements frequently enough. Again, acknowledging the team and the takeaways. If nothing else, I hope you take away from this that the neurological symptoms are almost always genetic. So doctors are somehow behind the curve on this. The parents know this, and we should somehow inform as many doctors as we can. AI is becoming, ubiquitous, and it's really good for rare diseases because it's incredibly deflationary as a tool, and the economics of rare disease has always been the biggest barrier to therapies. And so AI at almost every single stage of the pipeline of drug development, from the literature review to the discovery to the development to the clinical trial to just clinical care and practice, is it's entering everyday practice. And I hope that rare disease parents, advocates, researchers can leverage the productivity and scientific gains that are now available, and find the most creative ways to apply it to our field and our children. Yeah, our partnerships were critical. Our partners, all of the people who were part of Team Leo, we couldn't have done this without any one of them. And the standard of care needs to be better for rare disease patients. Standard of care does not cut it. And again, this is where AI can outshine human doctors and human knowledge by so many orders of magnitude, that I think it's enough. The change in quality is enough to overcome the inertia and the other barriers towards adoption. Being this end-to-end process, so we know we have all of Leo's patient information thanks to HIPAA. We have all of the costs of his care, and this was the cost of standard of care before the trial, right? So this is what was billed to insurance, and this is not even including any of, say, the lost work time for the parents. This is not including the reduction in quality of life. Leo, had he been a healthy baby or treated earlier, he could have been a productive member of society. That is an economic cost. There's all this therapy that the state provides him. There's caretaking for probably the rest of his life. And so these are, when you add this up, it comes out to millions, maybe $10 million. I don't know, but it's a number that is not so much less, maybe the same as the cost of the drug development program. And so, when we as a society and our representatives and our policymakers and all of these people think about this, maybe the economics of creating drugs one at a time, one patient by patient, they're not so far off from break even. And if that's true, then why aren't we doing it that way, instead of dumping a lot of this money into anti-epileptics and all of these these specialist referrals that ultimately really did not have any impact on his quality of life. And last, so I am a mathematician and a machine learning engineer. So this first principles concept, trying to do things from first principles, this is kind of a core part of who I am. And that is how I went into this project when he got diagnosed. So all of these are kind of, if you were a scientist and you wanted to solve a problem, you might not start with, well, this is how things have always been. You might start with, what are the constraints? And are there any constraints? Are there any laws of nature that prevent Leo from being dosed in 2 weeks? No, there aren't, right? What are the constraints? There are regulatory constraints. There are operational constraints. There are economic constraints. But these constraints can be overcome in ways that laws of nature cannot be overcome. These are human constraints. And we as fellow humans can work together to remove these constraints, so that in the future, one day, maybe in the near future, these children can be treated within weeks of being diagnosed by a drug that is safe and efficacious, and the whole life can be recovered and rescued. And that is a vision that I deeply believe in, and would like to work and am working towards. And technology and the advancement of science will make this future possible. I think that's it. Yes, that's it. Thank you so much for your time.